there's a really high complexity in the wiring up of the nerve cells. So that could be for doing high computation. But then all of the cells would be operating using what's essentially a binary code. So I called it a machine code, which not everyone likes, but I think it's the kind of like every single cell in the body would be operating with this mechanical interdependency between all the cells, which would be at the base level, would be at the binary level of these switches.
So hello and welcome to the Sheekey Science Show, where in this video I spoke with Ben Goult, Professor of Biochemistry at the University of Kent, where his lab researches the mechanical basis of memory and the role of the protein Talin as an information storing molecule. This Ben refers to as the MeshCODE theory. We discuss this theory of memory and more in our discussion. Enjoy. So hi Ben, thank you for joining me today and welcome to the Sheekey Science Show.
Hi Eleanor, thank you. Yeah, it's great to be here.
Thank you. So actually, I came across your research first, probably back in 2020. And so I feel bad that it's taken me so long to finally reach out to you to actually get you to come onto the channel because your MeshCODE theory is super interesting. And I'll be really excited to get onto that later in this discussion. But I thought just to begin with, one thing we should sort of discuss, and I'll sort of just jump straight into the question, what is mechanobiology and what is this whole subject area about?
Okay, so mechanobiology is an exploding area of biology and a lot of realization over the last decade or so that all the cells in our body and all the cells in all animals and all cells in all organisms really are very sensitive to physical forces, flow, different pressures and different movements. So basically mechanobiology is trying to understand how forces impact on biology and the different ways that cells can sense and respond to them, and how that can ultimately be important for the formation of higher orders of complexity, because each cell can respond to different mechanical signals. So that's probably what it is.
And so obviously that combines both like biology and physics and probably a bit of engineering. So what is your background and how did you get into this field?
Yes, so I got into this, basically it was an experiment which we did over a decade ago, which got me into mechanobiology, but prior to that, I was a biochemist. I did a degree in biochemistry, and then I did a PhD in drug discovery and approaches to measure drugs binding to proteins. And then I went back, and then I did my postdoc, which was actually on postsynaptic density and PSD 95. After that, I went into industry for a year and a bit to work on drug discovery. But then while I was doing my drug discovery at AstraZeneca, one of the things I was tasked with was to test all of the different NMR software packages and choose which one was the best and which one they might adopt. And this sounds a bit esoteric, but one of the new ones, the state-of-the-art ones, was a program called CCPN. And the guy who was making a lot of the scripts for it happened to work on Talin. And he sent me protein data and these scripts. So I spent the last 6 months of my time at AstraZeneca working on Talin. And it worked really nicely. And then he said, oh, you should come and have an interview. So in 2006, I actually came back into academia to work on this protein called Talin, which I assume most of this interview is going to be talking about. And since then, I've just had the most amazing journey following how Talin functions, what it does, starting from this very basic view of it just as an adapter protein up to where we are now of this really complex mechanical protein. And I was a straight-up biochemist, structural biologist, until we made a discovery where we had a region of Talin, one of these domains, which was doing a different job. It had two things it did, one when it was folded and one when it was unfolded. So it had to change its shape as a function of force, was the hypothesis. So it got stretched and then it did a different job after that. So we found a switch. And then after that, I got in touch with a guy called Professor Yan Ji in Singapore. There's a whole mechanobiology institute in Singapore. And I said, we found this switch. I really want to know how hard you have to pull to open this. And then that just set us on this journey with me and G, just exploring what Talin did and got us to where we are today. So that's how I got into mechanobiology, just from as an experimental finding of two different ligands binding to two different proteins which bound to the same small part of the Talin molecule in different states.
So yeah, exactly. As you say, we're probably going to be talking a lot about Talin. So just to keep get my viewers up to the same level, how would you describe Talin? Like, what is its structure, and how does that fit into like mechanosensing of the cell? Like, what does it mean to say an adapter protein?
No, that's a very good question. So I should have said that. So when I first started with Dave Critchley's lab at Leicester in 2006, it was well established by that time that Talin was the part of the adhesion complexes which hold all our cells in place. So on the edge of all of our cells, there's these receptors called integrins, and these can physically grab onto this underlying extracellular matrix. So all of our cells in our body attach onto this matrix, and that provides instructions on how they should behave. And the integrins on the inside of the cells are bound to these proteins called Talin, and Talin connects the integrin and the extracellular matrix complexes to the cytoskeleton, this meshwork inside the cell of force-generating machinery like actin, and so Talin sits there. So when I first started on it, Talin was relatively little was known about what it looked like, and it was just considered to be an adapter. So one of the first tasks we did was to work out what the molecule looks like, and that's how we discovered that there's 13 of these, what we now know are binary switches in there. It's got 13 of these folded helical bundles, which are the switches, which are the idea for this theory. And what was interesting was we were working on this, and it was just considered to be this adapter. But it was really strange because it's completely the same in every single animal, right down to slime mold, and every single animal up in evolutionary complexity. And over that whole evolutionary time, it's always had exactly the same domain structure, and they're all in the same order, these switches. And a lot of the residues, a lot of the amino acids which are important in us, are perfectly conserved in the slime molds in the soil in your garden. So clearly this molecule appeared and then has been maintained all the way through animal development and animal evolution. So that's what Talin is. It's this essential adapter. And it is essential because if you don't have it, it's lethal, and there's not many mutations which are viable where the organism develops correctly.
And so Talin is expressed in all cell types. Is that right?
Yes, there's two Talins, and Talin 1 and Talin 2. Talin 1's quite broad, it's in all of our cells. Talin 2 is also in all of our cells, but Talin 2 tends to be enriched in heart and in the brain. So they've got slightly different where they're expressed, where they're found. But yeah, it's in all cells.
So as you've already mentioned, you've spoken about these different domains and how they have this like unfolded and folded states. And yes. I'll probably in post editing put some pictures to sort of explain what this really means. But when you go from this folded to unfolded state, there's this massive increase in length that can be generated.
Yes.
And so that sort of led on to this MeshCODE theory, which you written now a couple of articles on. And so I was wondering if you could explain how you went from just looking at the structure and this sort of mechanosensing of this protein to having this like theory for coding binary information into memory.
So there's a bit of a step there, and it was like a conceptual change in my thinking. So what I'd say is for explaining what these switches look like, the animations which we've now made, it'd be very nice if you could show those, because there we physically show what this opening and closing of these domains looks like. And the opening and closing of the domains, the switches, the flicking of the switches, that's all experimental. We can show that at different levels. At a single molecule, we can physically manipulate these molecules. So we can show absolutely experimentally and definitively that these switches exist, that they open and close independently. And we can also show that they've got this key point called molecular memory, because if you stretch a Talin molecule, so you get a certain pattern of, so we call the folded domains 0 and then the open domains 1, just because there's two states, there's a closed and an open. If you have a Talin which is in a certain pattern and you hold it, it will stay in that pattern for minutes in our experiments. And if you have things which stabilize those patterns, then they can be very long-lived. So the idea of mechanical memory, we sort of we kind of discussed it back in 2016. But the real change in the thinking was, if you look at a cell in culture, so a lot of cell biology is done by looking at cells under a microscope. In there, these adhesion structures are quite dynamic. They only last, like, the turnover of the molecules is tens of seconds, so they're quite short-lived. But the idea came that if these cells become ordered, like in our brain, I don't think the adhesions are constantly assembling and disassembling every 10 seconds. I think they become ordered. And that higher ordering of the molecules led to the idea that these switches, these patterns of information, could be more persistent, and they could be much more ordered and more specific than what we currently imagine. So that change in thinking came from a very simple idea, actually. And it was when I was in Australia. I'd just been to give a talk in Sydney. And a guy there called Peter Gunning, he asked me a question. He says, so you show this nice Talin unfolding and refolding, but that's just one Talin molecule. There's 100 in a cell. How do they all work together? And I was sat at Sydney Airport and trying to think about it. And then it was like, the only way you could bring that together is if the system is ordered, if they're all working together. And then if they're all ordered, then you can have much more organization. And I think the organization of the cells is probably much higher in our body, and particularly in our brain, than what we currently think from the limitations of trying to physically visualize a cell in culture, which is already quite complicated. So that was the big step, because then you could have an idea where if you've got a cell which is moving around, then the forces experienced by each Talin molecule are in part from outside, the movement, and also from inside, the motors and the fourth generation machinery. However, if the outside is relatively constant, if the cell makes its ECM nest perfectly, then that becomes constant, and then the cell can control the switches from inside by switching on its motor proteins and pulling. And it was that step change, because then I thought, then the cell can be in total control of that, and that led to this massive idea from that initial thought, you could write data into these adhesions, to, these adhesions are essential for holding every synapse in the brain together, what if these synaptic scaffold binary switches were a way to write information, but also coordinate the activity of that synapse. And maybe we'll get onto that. That's in the latest story. Let me just turn off my WhatsApp because it's pinging. I don't want that. So that's the general idea. That's where we got to. Well, I got to where we are, and that's the general basis of the MeshCODE. That there's a meshwork of binary switches in all of our cells, but in a higher ordering inside the synapses of our brain, where these nerve cells are talking to each other via these connections.
Yeah, so I've got several follow-up questions to that. And so just to sort of summarize, to make sure that I've kind of understood this correctly, you can somewhat, as we've described with Talin, having these folded and unfolded regions, some sort of mechanical spring within a cell attached to both actin filaments within the cell, but also connected through integrins to the extracellular matrix outside the cell.
Yes.
And so in theory, if you had forces from either side, you could sort of stretch it from both directions. But you're saying that within the brain, or maybe with other tissues as well, there's more of a fixed force or continuous sort of sensing from the extracellular matrix, that the Talin is responding to internal signals within the cell. Is that correct?
Yeah, so so that's very nice, succinctly described. But yeah, the idea would be that these synapses, because you've got say a dendrite, and you've got these little spines sticking up, and then they've got their little bulb on and they're talking. These are isolated mechanical units from the rest of the cell, because they've got this long shaft and then this thing. If you think about the Talins on the bottom of the cell, then they've got lots of different variables and different things. But the cells, the brain, is well established from lots of stunning work looking at ECM maintenance, that they invest a huge amount of energy into making the ECM how they want it to be. And there's loads of things where that goes wrong. But then if the ECM becomes so the cell knows what it's like, then it doesn't mean it's not changing, but it means that the cell, when it pulls, it's a predictable outcome, which can be generated from within. And what's been shown by other researchers is that the synaptic transmission, where the synapse, the presynaptic releases the vesicle into the cleft, and then that's sucked up by the postsynaptic. That's well shown to increase changes in the cytoskeleton and switching on of the motor proteins in the postsynaptic side. So what the idea here is that these well-established things, which are being shown by the neuroscientist community, they would be orchestrating the operation of these Talin molecules and these switches in a way that you could actually capture these events in a quantized state, like by flicking a switch. So that's the idea.
So what are like the downstream functional consequences of having Talin in these different folded and unfolded states?
Yes. So that's another very good question. And what was shown not a long time, it's all relative, I suppose, but 15 years ago from Adam Engler and Dennis Disher's lab was that they could take a stem cell and put it onto different different surfaces which were coated with matrix. And they could get that stem cell, which is undifferentiated, to differentiate into lots of different lineages. And the trick that they did was they kept everything constant. The only thing they varied was the stiffness of the matrix this cell was sat on. So if they sat the cell on jelly, it would differentiate and become a fat cell or a nerve cell. And if they sat it on a matrix which was on glass, it would become a bone cell. So what's happened is that those cells have grabbed on, they've interrogated what they're attached to, and then that's ultimately led to this process called mechanotransduction, which is where the cell has converted those mechanical forces and transduced them into changes in the gene expression and the behavior of the cell. So the idea in the MeshCODE theory would be that would be happening on a much more complex, well, not more complex than that, but each of these synapses would be updating, and that would have an effect on the gene expression and the mechanotransduction of that neuron. And because each synapse would be getting signals from different neurons, then they would be talking to each other in this way by a signal coming from one cell to the next cell, and then that would do a calculation, and then that might trigger to another cell, and then you could get this whole circuit which would be being updated by this mechanotransduction in a hyper-ordered system, in a perfectly ordered system, I try and argue, but and that would be the general idea, I suppose.
Yeah, because in terms of like thinking about memory and synaptic transmission, there's often a lot of talk about the strength of the connections and that being linked to like memory formation. But I guess from your theory, it's more about the speed of the downstream consequences following the synaptic connection is more encoded into how the Talin is structured.
So yes and no. So what I think, and I think one of the strengths of the idea is it's consistent with the current thinking of this electrical and chemical signaling of the brain. That's taking place. But what would be happening underneath the hood would be this mechanical computation where that electrochemical signaling would be coordinating these motor proteins to change these patterns of information written into the shape of these molecules. Because that's kind of what I'm saying, is that the information is written into the shape of these Talin molecules within each of the synapses. But one of the big questions we got I got, but then talking to lots of people, the common question was like, how would those changes in this binary pattern actually change the activity of the synapses? Which is why this new paper, which came out 2 weeks ago, we started to try and understand how would these changes in the shape of molecules change the signaling and change which synapses have got high activity and which have got low activity and all the variations in between. And there we started off when we were trying to build a simulation of Talin mechanical response, which was a bit ambitious in retrospect. But within about a week of trying to do that, we spotted something much more simple, but much more obvious and much more important, I think, in that, as you said at the start, these switches opening, they, as well as changing what ligands bound to that switch, which proteins are on it, they also introduce a big jump in the extension. So a domain, when it's folded, is 5 nanometers. When it's open, it can stretch as far as 50 nanometers. So it's got 10 times longer. But the main implication of that is it's got 10 times longer. Its signals changed. But every single switch after that has also moved, and the active cytoskeleton has also moved that far in. So what we get to is we get this idea that these And the best example we've got of this, in fact, I'm saying this a bit back to front, is we've got a whole series of enzymes which sit on Talin on these switches. So enzymes have substrates which they either phosphorylate or they acetylate or they change. They provide a chemical change to. And these enzymes which are sat on these switches have substrates within the synapse. So if you've got an enzyme and a substrate right next door to each other, then this can easily modify that. But if you physically hold them a long way apart, like if the switch in the middle had opened and these are now held apart, then the activity is really, really low because this can't actually reach to that. So the idea would be that there would be patterns, there'd be codes, combinations of these switches where all of the enzymes would be lined up, so you go duh, 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 duh. And then other ones where they were held apart, which would have very low activity. And that was the idea in this new paper, well, not the idea, the realization that this organization of molecules in the Z dimension, so you'd have how do I do it with my hands? You have the synapse here with all the Talin molecules attached coming down. Then if this is the X and Y dimension, then Z going away, like down the synapse, you'd organize all of the enzymes and substrates in this Z dimension. So you'd have some which would be really efficient and really active synapses, and then some which would be have low activity. And if you looked at them with a microscope or you looked what was in them, there would be relatively little change because it'd be the same molecules, they'd just be organized differently. And they'd be organized based on the signals that specific synapse had received. So that would be how a binary code would convert into a regulation of a neural network. You'd have these synapses which could be dialed up or down by these signals it's received, which change these codes.
I see. And so actually one thing you made me think of is with what sort of types of memories in terms of, you can talk about short-term and long-term memory, but also given that this is like a protein basis for like where the memory is potentially encoded, proteins have a sort of like turnover within a cell. So how does like protein turnover, like what is the turnover of Talin and how does that sort of fit in with your theory?
And that's an excellent question. And that's been the major limitation on the consideration of biomolecules as molecular storage in biomolecules, because molecules have a turnover time. Obviously, if a molecule is tethered to the integrin at one end and the cytoskeleton at the other, its turnover is a lot less. But once we drew these molecules to scale, then what we could see is that you could easily imagine that. So maybe I'll just back up one step. So the general update to the MeshCODE theory, which came from this new MeshCODE to scale paper, was the idea of that within each synapse there's there's three parts actually, two ordered parts. One of them which is permanently attached to the synaptic cleft, and then a mechanically operated part which could be moving up and down. And it'd be moving up and down because of all of these Talin springs, as you so nicely put it, which the cytoskeleton would be, the actin would be, the motors would be pulling up and down. And in which way that it's quite a large part of the machinery which would be moving. So if one Talin was to ping off and get turned over, then the whole machinery wouldn't suddenly collapse. So it'd be like if you were driving a lorry and you got a flat tire, not you were driving a lorry, probably not, but if we were driving a lorry and one tire burst, you wouldn't just suddenly veer off into the ditch because it would keep going on the others. You'd have to stop and change it. So the idea came that maybe if all of these Talins are coordinating this mechanically operated part of the machinery, then it wouldn't be explicitly dependent on every single molecule. So that would allow for turnover. And it was actually Francis Crick in 1984, I think, who said that this was why it's hard to imagine protein-based memory because of this limitation. But I think this overcomes it. And that was because when we could see what these molecules would look like, it led to this idea that they might all be working as a collective, as a machinery.
Okay. I like how, I mean, obviously I understand like how some proteins get degraded, but do you know how Talin might get degraded if it is, like, as you say, attached? Is it just wear and tear, or like what physically, is there any regulation over its turnover?
I'm sure there is. So if you measure the turnover of Talin in cells in culture, then there's two distinct populations. One bit which turns over quite fast in like minutes, and then one which is more stable for tens of minutes. If you do the same experiment, which was done by another group in fruit fly, in the wings or the muscles of fruit fly, then what you can see is that the turnover of the Talin molecules in the muscle is much longer. It's on the days scale. So presumably, if it's a proper ordered system, it could be much longer than that. But all molecules will turn over. Like, that's part of how you maintain the integrity of the cell, and that's why when aging, when you start to get defects in that can cause problems where you get accumulation of waste products or damaged proteins.
Okay, yeah, and so like to continue your idea about timescales, and obviously we've already mentioned earlier that Talin is expressed in different cell types. We obviously, when we think about memory, we often always think about the brain, but if Talin is being expressed along with these other proteins involved in this MeshCODE theory in different cells, do you think that there's some element of memory in other cell types? And by that I mean not like memory is how we might think of it, but like how a cell might remember how to respond to a certain stimuli, or do you think it's universal property of cells as opposed to it being a unique thing that we just see in synaptic connections?
Yeah, so that's an excellent question. So yeah, I think it's definitely a universal property of cells, and I think the organization of the cytoskeleton and the switches at the bottom of the cytoskeleton would definitely be, in all of our cells, would be information carrying and could be updated by a contractility, like by switching the motors on. And one of the ideas I speculate in the paper is exactly that. Like, a lot of the synapses are beautiful for doing computation, because you can send a signal and change that MeshCODE, which then would update that neuron. So you could do very complex, because there's a really high complexity in the wiring up of the nerve cells. So that could be for doing high computation. But then all of the cells would be operating using what's essentially a binary code. So I called it a machine code, which not everyone likes, but I think it's the kind of like every single cell in the body would be operating with this mechanical interdependency between all the cells, which would be at the base level, would be at the binary level of these switches. So yeah, I think all of the cells, and that's definitely the case, because all animals, even going down slime mold, slime molds have been well established to have a basic type of memory where they will map out where food is and go in a direct route. And so I think that's an intrinsic, and they've got the same machinery, the MeshCODE machinery. So I think it's an intrinsic part of what makes a cell. So yes.
Yeah, it's interesting.
Yeah.
And so, like, obviously going on from that, and obviously studying things in human organisms is very hard to do. But like, what's sort of the best evidence so far about sort of testing or proving that this is where the memory or the information is being encoded? Obviously, we have the structural studies where we can see that there is a way of encoding this information potentially, but is there a way that you can sort of tweak it or rearrange the structure of the domains? Or like, how would you, what to you is the best evidence you have so far?
Yes. So again, excellent question. So for sure, we can show on the single molecule, these are memory molecules, we can show that the integrity of synapses and the wiring up of nerves to form these synapses, that's Talin dependent. So the talin's an integral part of the scaffolding around the synapses, and it's a memory molecule. Obviously, when you have those two, there's a join up in the middle where is the memory part of the molecule at play in the synapses, and so that's the bit we're desperately trying to test. So we're trying this in multiple different ways. I think the best evidence we've got is some work we're doing in collaboration with a lab in Yale, with Tony Koleski, where we're actually measuring the length of these molecules within dendritic spines. And we can clearly show that the Talin is very much longer than what it would be if it was not stretched at all. But most excitingly is that the different spines on the same dendrite have got different lengths of these Talins. So the big thing there is if we can correlate, so it's changing its shape in the different ones. If we can correlate that to the activity, then that'll be the jackpot. And we're trying to do that at the minute. Because if we can show that the activity both changes and the length changes, and we could get predictive on that, that would be really important. So that's a work in progress. The other data we've been getting is trying to work out how to visualize these different shape changes. So make probes which can recognize whether certain switch is in a 1 or an O state and things like that. And then we've also been using a technique with a group in Sweden where we can actually look at which proteins have changed their shape. So you take two sets of neurons. You take one set of neurons, you split it in two, you stimulate one half, and then you submit them both for this technique where you look for domains which have changed shape. And it's a type of mass spectrometry technique. And what you can clearly show there is that the stimulated and the unstimulated, they have different patterns of switches. So all of this is kind of slightly tangential from each other, but I'm hoping if we bring them all together and we can correlate them all, then that would be good. And the other aspect we've got on the experimental is we've got a series of neurodevelopmental diseases linked to mutations in a few of them are in Talin, but most of them are in the proteins which interact with Talin. So if we can join that up and show that these mutations are caused because of a failure of the mechanical regulation. So I hope in 2023 will be the year where we can actually show this, because we can see it now. I could physically show you images of these proteins changing their at different lengths, but we're still not quite nailing it yet. So getting there.
Yeah, so one of the things I was going to ask you about was mutations and if there's any links with diseases. I say, like, is Talin commonly mutated? Is it like rare but lethal, so it's not very common?
So up until last year, I thought there was, or maybe 2 years ago, I thought there was no mutations in Talin. And now with the huge genomics, there's a few which have very specific diseases. Interestingly, in Talin 2, the other isoform, there's no mutations whatsoever. So yet identified. So it's obviously very well conserved. But the things which interact and decorate Talin, there's lots of mutations in those. And in these adhesion complexes, there's hundreds of diseases caused by mutations in them. But Talin, right in the middle, is so important that they're very few and far between.
That's interesting. And so, like, in terms of the genetic structure as well, is it, you mentioned there's Talin 1, Talin 2. Is it that there's just one copy of each gene, or is there gene variants?
So the Talin 2 can have some so there's two copies of each gene. So that there's no homozygous mutants of Talin where both copies are messed up, but the heterozygous ones where one copy of the gene has got a mistake, then they have disease. But the two Talins are separate genes, so there's no mutations in the Talin 2 gene, which I'm aware of. And that's also the one which is more brain expressed. So one idea is that the two have got very similar, but then just subtly different roles and slightly different localization. That's something else we're actively exploring.
That's interesting. And like also, because I forgot to ask earlier, when we spoke about there's 13 domains of Talin. So what is like the maximum, in terms of this binary code, amount of information that could be implemented? Because is it more complicated because we have multiple Talin molecules within one?
So I think this is a good question, and I'm not 100% sure. Like, we know that the third domain will always open first. Or in every experiment we've done, it's the weakest domain. So that one's always going to be at the start. So there's a slight ordering to them because there's some which are really strong, some which are very weak, and then different ones in the middle. So there's not 2 to the 13 permutations because some of them won't happen in that order. Trying to get to the total number of bits of information, you could speculate based on how many switches we think there are. And it might be, I don't know is the very short answer, but it's definitely not 2 to 13. It's probably less than that.
Okay. And like also, because you mentioned it earlier, like the fact that not only is Talin conserved, but the order of the domains is also highly conserved, which I guess, is in support of your theory that whatever this is doing is clearly very important.
Yes, because I think there's meaning to that order, and I think the order at which they open up is instructive of what the change in the signaling at that site and the change in the length of the molecule at that site. and it must the meaning of that is quite and so something which we're trying to do with in-house is to try and understand the rules of computation on a single molecule, because we can show that these switches are opening, but could we actually answer those questions? How many states do you get? What patterns do you get routinely? And try and, because there's also a possibility of using these molecules for computation in like a machine or in something else to actually harness it for computation in a biocomputer or something. But first, I think there's still a lot to do to understand the rules of Talin computation, and that's a work in progress. Yeah.
Okay. Yeah. Because one thing that I think would be interesting would be to see, because if it's lethal to have actual mutations in Talin, but if you just sort of switch the order of the domains, would that affect memory or some sort of specific function within the organism?
No, that would be great. The problem is because of so in cells, people have, like, one of the things which we've also been doing, which is similar to that, is that we can, because we know the structure of this, because that's the first thing we did, we know what each domain looks like where every single atom in that domain is. And what we've been able to do using that information is to make changes, mutations, which stabilize the switch or make the switch have a hair trigger. So that's a way to sort of change the order without changing the order, because you could make the domain which opens first much stronger, so it doesn't open first, it opens second, or things like that. And that's been experiments which have used those sort of mutations. That changes how cells in culture behave. So that's one way to do it. And on that note, some of the reasons why these switches open and close is because they've got, inside the core of them, inside the middle, they've got residues called serines and threonines, which are actually where the phosphorylations occur. So enzymes interact with those. So one of the things which is also very interesting is that the enzymes change the switches and change the order which they'll open in. So there's a lot of this going on, which the cell's doing intentionally. My concern was if we tried to reorganize this, I think maybe on the cell in culture it'd be all right, and that's something we should definitely try. That's a good suggestion. But in a more complex system, I don't think it would. I think the computation would fail. It would trip over because everything would become disorganized.
I see. So it's likely to be lethal, as opposed to affecting like a specific memory function. Like it might affect all cell types.
Yes, and I think there'll be because developments and there's a lot of mechanobiology going on at each stage. The next stage is based on where it's already at. So if it falls over early on, that's usually lethal, because when it goes to do the next stage, it's out of sync, and in this idea, then it would just, the mechanical calculation which is causing the development of the organism would have a catastrophic error, it would just fail. It would fail. So it'd be good to try though, and certainly try it on a cell in culture or a neuron in culture, which should be very doable. Yeah, I think that'd be good.
Yeah, I think it'd be really interesting. And so with memory as well, there's obviously lots of different theories about where memory is encoded, how is it encoded. Based on some of these studies where we know Talin's really important, it somewhat suggests that Talin seems necessary for whatever memory is, but clearly by itself maybe isn't sufficient. Or do you think there's redundancy in the basis of memory, or I don't know, is there a backup or proofreading or alternative? It's just Talin's part of the story, but it's not the complete picture.
Well, Talin's part of the really complex mechanical machinery of the cell, and it's essential for that, but it's not working in isolation. All of the things which regulate that, all of the density of the receptors and the vesicles being released, they're all essential. And it's not just a single synapse in isolation, they're all linked together. And there's a lot of work on neural networks and these circuits information, where the problem I always have with some of those ideas is that it's basically the difference between actually writing something on a disk and holding it in short-term memory and RAM. If you're trying to hold all the ideas simultaneously, then that would take a lot of energy, be very complicated. Whereas if you imagine the neural net, where at each node you've got a set of switches where you can change that node and capture it in a really quantized, specific way, then a reimagination of the neural net idea. Because I think the idea that synaptic signaling and synaptic strength is reasonably well accepted. There's people who challenge it, but I think it's quite well established that changes in the transmission through synapses is how information is stored. And here that would be written in a more complex way, not just a simple on or off. It would be like there'd be more information written in each synapse, and they would be each node in a complex circuit. So it's definitely not sufficient on its own. And if we have a Talin molecule in our test tube, it very limited the number of information we could write into it. But as part of the machinery and as part of this complex, beautiful cell-cell signaling which is going on, then it's a central player, I think, or the theory would suggest.
Yes, one thing actually you made me think of just now is like the efficiency of the system. So obviously things like DNA replication, protein synthesis, all very highly energetic processes within a cell, and even like synaptic electrical transmission with the sodium ions and potassium pump, that being extremely energetic as well. Yeah. In terms of like how the Talin is like stretched and how it gets like fixed in that position, or how it changes. I'm imagining like a spring now in my head, but obviously like that's to some extent it's maybe passive, or like is there what energy is required for this process, and is it therefore linking back to how the brain is like super efficient in terms of memory isn't something that uses a lot of energy? I mean, as much as I think like revising for exams is very energetic, maybe like a lot of memory storage doesn't actually need energy. It's like just based on sensing. I don't.
No, that's an excellent question. That's where I think one of the strengths of this is, because in fact memory molecules and things which decorate them, they only need like a basal level, a low level of force, like intracellular tension, to store this information. And then to change the signal, you have a temporary increase in tension. So you switch a motor protein on, it pulls until the next switch opens, and then it goes back to what's a low energy state. And these patterns, because they're not just a molecule on their own, like things are sat on them and fixing these patterns in these states, the energy is only required to update them. So when you have a synaptic signaling event, that would update this pattern. And the rest of the time, it would be held at a low energy. So I think that this is one of the big strengths, because also going back to the neural net, if you're constantly trying to keep everything in RAM, you'd need a lot of neural activity to maintain that. Whereas here, the default resting state would be a low energy state, not a no energy. You need low energy for basic upkeep of the cell. And then the signals would be a transient increase. So I think it's a much lower way to consider memory, I would say.
Okay, yeah.
You just have an input to change the pattern.
And so given that, obviously, like how this theory is described as being like a mesh code, like it is like literally a mesh, in terms of like potential ethical issues about how if we somehow determine the molecular entity of what memory is, and like links with like memory manipulation or memory changes, do you think that would even be possible given what we've described, like how it's encoded and fixed, and like what are your thoughts on that?
Yeah, so I really like the question, and I think this is where you can really mesh up against science fiction, because of the implications of it. So the MeshCODE theory is predicting that there's a physical location for all the information written into the shapes of these molecules in the synapses of our neurons in our brain, and they're physically written to specific locations. So that obviously leads to the implication that potentially you could, if you understood how the code that they were written in, then you could read that information. Obviously, we're nowhere near that. We don't even prove that there's a code. But if you do work that out, and with like SSD cards, like electrical storage, on like the little terabyte chip which will fit on your fingernail, like what we use in our phones and stuff, there the information is read back by the electrical signal and the route it takes back through the pages of information. So here, maybe the electrical fingerprint, because that's what people measure at the minute. They measure these electrical fingerprints of brain activity. If you could link that to what the underlying pattern was of information and extract it out, then of course, it'd be tempting to speculate that you could read the memories. And then the obvious thing is, could you then do the inverse and write them? And again, we're nowhere near that yet, but potentially. And then it also leads to, but then you need to understand what this coding is. It's a binary code, maybe. That's what I'm arguing. I think we're binary creatures. I think all animals are. Could you understand that code? And then could you understand, you'd need like to consider kind of the operating system, or how it's written, what format are we using? And you need to understand that to be able to retrieve the meaning. But I would say that's very possible, possibly, once you understand it. So yeah, and then you get into if it's a proper binary thing with a human operating system, say this is proper science fiction now, so this is detached from the bit we talked about, which is more grounded. But if there was an operating system with a specific, then could you upload that onto a computer? I know there's a lot of people interested in that. Could you preserve brains and recover the information? So then, as soon as any of that becomes possible, then there's vast ethical issues. But I don't think they will hit us probably in my lifetime, probably in your lifetime. But I don't know where that would be down the track. But yeah, there would be. But it definitely mashes up against science fiction and science fact. But that might be where we could do this.
Yeah, and like given that we have sort of realmed into science fiction a little bit, and speaking about like preservation, I think it was the other week I was making a video on cryobiology and looking at how memory, whether or not if you freeze a brain, would that memory still exist? And I guess there was like there's overlap in that and understanding if there's like a physical basis for memory. Yes. And so I think I can't remember entirely what the study was I was looking at, but there was something to suggest that like long-term memory was perturbed by freezing, but I don't know, there might be support for some having physical storage or not. I don't know.
Yes. So I accidentally got involved peripherally with there's quite heated arguments about this on Twitter. I found between people who think you can pickle a brain and get the information out. This is very simple. They will hate me more for this, but like the idea that you could preserve it with formaldehyde or some form of pickling, versus the ones who think you'd need to freeze it, and then you could, like the cryopreservation, then you could warm it back up and get the information back. And I would definitely be more of the you need to freeze it and then try and really get it going. But if you could get it so that these circuits would not conduct electricity, it's a bit simple, but get them so that they would signal and start talking to each other, then potentially it could be possible. But also the good... So a couple of ideas which lead from that in terms of how we might use this for something more useful is if information is written in the shape of these molecules in the synapses, then over time, if you get a bit of damage to a bit of the synapse, then you might lose that information and it might become unretrievable because the patterns of ones and O's have got out of sync with the rest of the brain. So one of the things we're very interested in is aging of the brain, but more importantly, Alzheimer's disease. Maybe you could imagine that these plaques or the thing ahead of the plaques is corrupting this information. Like these a certain set of synapses become out of sync from the rest. And then it'd be like if you have a CD and you scratch it, you lose that information. It's never retrievable because it's gone. So the idea that these binary switches might be getting perturbed by these drivers of these awful loss of memory, loss of mind sort of diseases is somewhere I'm quite hoping to make some ground. Yes, but again, that's kind of science fiction at the minute, but hopefully we've got some quite nice experiments and data which indicates that we're on the right lines in that route, but early days. Maybe you can have me down in a couple of years and I'll tell you all about it.
I don't know, I'd love to. And also, like, I was actually listening to a lecture online about the extracellular matrix this morning, and it was sort of saying, like, how under-researched it still sort of is. I mean, there is lots of research being done, but how much still there is to learn about it. But it was speaking about it in the context of aging, and obviously given Alzheimer's disease, where you have, like, these amyloid beta plaques, and also, like, glycation and, like, stiffening of the matrix in different tissues as well. And obviously, if we go back to what we said, like, way earlier, where the idea of this Talin response is that there's some sort of fixed, you fix one part in the extracellular matrix and it's like the internal response that's getting changed. If now we're changing both sides, like the spring's getting altered from both directions, then maybe it loses, as you say, this ability to keep the memory updated, and that could therefore link to memory loss with age.
Of course, that's a really great suggestion. And what's really going back to the stem cell, which differentiates as you change the stiffness, then if the stiffness is changing because of a disease or because of aging or because of less loss of the tight regulation of it, then that's going to change all of the mechanical signals downstream of that. And that's what happens in a lot of cancers and a lot of disease where you get a stiffening, you get fibrosis. And you can sort of imagine it quite easily with the switches, because if you were to try and climb up a ladder and the ladder was a rope ladder, then you use a lot of work because it's hard to overcome those to convert into an upward movement. If you try and climb up a wooden ladder, you can go as fast as, like pretty fast, at maximum speed for whatever your ladder climbing ability is. And that's because it's much easier to overcome these force thresholds with a stiffer. So these switches will open much more easily if the environment, if the matrix becomes stiffened. If the mechanical computation is assuming that it knows what the matrix is, and then the matrix is stiffer, then you will get the wrong patterns, you'll get the wrong switches opening. And that is definitely how that could happen in the aging brain. As it gradually loses its this super-ordered state, which we're predicting here, and it starts to get stiff in some parts, and then that's corrupting bits of data, which then get out of sync with others, and the whole thing. So yeah, I think that's a very good point. And the ECM, there's a lot to understand about that. It's really complicated.
Yeah, no, and I think, like, I mean, a lot of the sort of aging literature is about like loss of information or like loss of cell identity, and obviously, and like cancer and other examples, it all does sort of interplay with like the ECM and how like that's connected with, like as you mentioned, through actin to like the nuclear periphery and how that changes gene expression. And so it all kind of makes sense. And I guess the interest is like, how do we then either prevent it from happening, or if it's happened, how do we then sort of reverse it or like regain the correct stiffness for the cell?
I don't think anyone's ever going to live forever, because I think when you go through development, there's like a really beautiful ordering of it. And I think at the end of development, everything's mechanically synchronized and everything is all ordered together. And that's why you can spot beauty in young anything, because they're mechanically perfect, and they've got that radiant skin and everything's perfect. And then over time, as you get little bits of damage or bits of changes, they slightly get out of order. So this long range synchronization of a cell here to here, I think, well, maybe this isn't a very good answer, but I think that you gradually just spiral away from this high ordering of this system. And that's sort of like kind of impossible, because kind of unavoidable. This is not going to resonate well with the anti-aging people, but it seems to be kind of inevitable that you'll lose that long range order. I think you could slow it down, but as it spirals out of control and they all get out desynchronized relative to each other, then that would lead to aging. In the concept of neurodegenerative diseases, where you're actually losing that information, like in dementia and Alzheimer's, then maybe there's a route to slow that down, because you could stop those plaques from stiffening it, or if there's a toxic intermediate, you could stop that from affecting these codes quite as quickly. So there might be new ways to treat it, which aren't anti-plaque drugs, which don't seem to be going too good. But I don't think you'd ever stop it, because I think it's an inevitable fighting entropy, really.
Yeah, it's given me a lot to think about. I think the reality is, at the moment, I don't think we know enough to really argue either way, or like I think there's still more to learn. Hopefully there might be some hope, but to finding ways to sort of like reverse the damage, or see if there is a way of recovering the information.
Yes, for sure. I think slowing it down, and it might be, what I would like to think is, maybe if you spotted Alzheimer's coming on 10 years sooner or something like that, like maybe there's a marker, you can see that you have got this sort of loss of information, this accumulation of damage, or this loss of this coding, then you might be able to intercept it earlier and slow it right down a lot. Because I remember one of my teachers when I was at uni, he said, you don't need to cure cancer, you just need to stop it long enough until old age does what it does. Like, and same with the loss of memory. You only have to, if you could slow it down by 20 years, then that would probably be good enough. Unless we are going to live forever, and then it'd be like not. my feeling is that would be good enough just to slow it down and just to preserve it as long as possible.
Yeah.
And that's how I'd like to try and think we might make a dent in that.
Yeah, so I guess while that area is still being researched, one thing that I thought as a bit of a sidetrack was applications of this knowledge of understanding how Talin operates. I saw another recent paper where you were describing Talin as like a shock absorber and using it in like different materials. So, I mean, I haven't really explained too much, but would you be able to elaborate on like what's next in the Talin space besides looking at just the MeshCODE theory?
So that was a really nice side spin-off from what we were doing, because when we were stretching the single molecules of Talin and measuring Talin how it behaves in cells, you could clearly show that it serves as a shock absorber, because if you pull really fast, then these domains opening up introduce a lot of tension in, which makes the force drop, and then it starts to accumulate again, and then the next switch falls. So it buffers force really effectively. And we showed that in a paper in 2016. So each of these Talin molecules is a shock absorber. So then I've got a chemist friend downstairs, who Jen Hiscock, and we were just having a chat one day, and the idea came about. I said, we've got these shock absorbing proteins, and she said we could make a protein-based material where we take the Talin as a monomer, but polymerize it. So we link together, I don't know, trillions and trillions of copies of some of these switches, and to see whether you could capture that nanoscale shock absorbing on a material level. And the short answer was, it worked, and it worked way better than what we had predicted. And it was, these materials have got this, it really captures it. And it makes sense because you've got all of these switches which are doing a little tiny shock absorbing, but loads of them polymerized, like joined together into a big gel. And the nicest experiment there was what my PhD student did for one of his final experiments, where we've got an astrophysics department downstairs, and they've got one of these light gas guns which is longer than this room, which fires like space dust, they use it for, but tiny little bits of sand, basalt particles, at 1.5 kilometers per second. So we put our bit of gel in, and then we fire it down. And the cool thing was that the gel was still intact, but then when we put it under the microscope, we could see all of the projectiles embedded in this. So that was quite exciting to the physicists, because they want to capture this stuff to study it. Because this space dust, from what I gather from talking to them, is a massive problem for spacesuits and the International Space Station and stuff, because it's whizzing around the Earth and whacking into them, and you don't want it to go through the visor of the spacesuit. So we're currently doing a lot of work on trying to develop that to scale it up and to make that into a new type of material, a shock absorbing material, using what we know about Talin. So that's a side project of what I do. The main thing is, I think, is to understand the MeshCODE. I mean, that's my passion, and that's where I think we can make the biggest difference. And if we could prove that there's a binary code in animals, that'd be really cool. And the different diseases which are caused by, my dog has got epilepsy, and we've got a pretty well nailed down link between mechanical signaling and epilepsy now. So if I could help contribute to epilepsy and what we know about epilepsy, that'd be really nice. And besides that, just to carry on understanding this molecule, because it's so amazing. And I've spent the last 15 years working on, maybe even longer, working on this same molecule to try and understand what it does. And every time we do a set of experiments, you suddenly can see a different set of what it's doing and how it's operating. So I think the main goal of my research is to carry on working and understanding Talin as this memory molecule in all animals.
Well, I hope you do, because I mean, what you've published so far is super fascinating, and I'm keen to see some of these experiments come out. So just to kind of I guess, wrap up our conversation, I was just wondering if you could give any sort of advice for maybe younger scientists. Obviously, you've now got a very impressive career and done some really great research. What do you think were sort of key values that you sort of took along with you?
A really good question. So, to persevere, I think it's really hard, and to be persistent and to work hard, I think that's a given. And to be nice to people, because I really try the best thing I like about this game is collaborating with people and collaborating with people who are really good at what they do. So I know the bits I can do, and then collaborating and building these personal connections with people. I think that's really my number one tip. Like I think it's Google's motto of like, don't be a dick. I mean, it's true. Like, just be nice and be open. And I try and be as generous as I can, and I try and be fair in collaborations. It's not about winning on something, because we're trying to do something much bigger than that. So I think to work hard and be nice and to just, there's a lot of ups and downs. And I had a very circuitous route till I got onto Talin, because like when I was doing my PhD and I had my supervisor, sadly he died in a car crash. So then I had to move and change project. And then my first postdoc we got scooped on, and that's the fear of all scientists, I think. But and then so I had a very, very slow start. It was like, I don't know, 4 years after my PhD when I got my first paper. And then going into industry, learned a whole new set of skills to be organized, because I wasn't very organized before, and I'm slightly less disorganized now. Ever so slightly better than I was before. That's probably my advice. Just be nice and work hard.
I like that advice.
And a lot of it is thinking. I spend a lot of time thinking about the same thing, for better or worse. But
I mean, do you read quite widely?
You've obviously really well read. It's been very impressive this. So I think read, and also go to seminars, because we always have a problem drumming up everyone to go to them, but seeing stuff which isn't in your field is really important. Like we got £1 million grant off a talk where we went to look at something on amyloids, which I don't work on, but this missing piece of the puzzle, and then you write it and you suddenly. So I think going to seminars is a good tip as well, because even if it's not a very good seminar, you learn about why it's not very good, or you always learn something from a seminar, I think. So.
Yeah, I should probably go to more seminars. I'm pretty bad.
And it's always like, oh, I'm so busy. But especially if they allow you to eat your lunch in there, then it's almost like a freebie. And
that is true. They give us pizza now. There's no excuse.
Yeah, cookie at the end, and then you'll be there. So and that may not be very good advice, but that's probably what I think.
No, I think that is good advice. And do you have any specific books or anything that you would recommend people to read?
So I read there's a really good book on cell biology by the numbers. I've got it up here somewhere. I think this one's amazing.
Oh, I added that to my Amazon basket literally last week.
So that's a good one. And then there was, when I first started getting into mechanical memory, and I started to Google and look into it in a bit more serious, moving away from looking at cell migration to starting to think about memory. There's a fantastic book by, I forget the first author's name, but Randy Galistel, and it's on the computational mind. And it doesn't provide any answers, but it addresses the need for a read-write memory in animals. To be able to do computation, you've got to have a read-write memory. So I read that. I just read it on flights. I don't read it the rest of the time, but just read it when I'm flying, and learned a lot. And it sort of helped to formulate the ideas I had into a framework which made sense. So that's another really excellent book. Yeah, those two, I'd say.
Oh, great. Okay. I will definitely buy that book at some point.
You should get it. Yeah, ask for it for Christmas, because it's well written, and it's just fascinating to just think about how many molecules there are, how long they are, because they don't really talk about it in there, but drawing the MeshCODE to scale, which is in our new paper, that's where we suddenly could see why that would work. And also to see that these molecules don't look like what they're drawn in textbooks, where they're all these nice, pretty little circles. It's very good for communicating an idea, but it's not so good for actually understanding what they might be doing, because it's not accurate.
Yeah, that's completely true. I'm kind of guilty. Yeah, I should read this book basically.
Yeah.
So where can my viewers go if they want to learn more about your research or get access to some of your papers?
Say that again.
Where could they go to find out more about your research and your publications?
Well, I would start with either the MeshCODE papers. If you Google MeshCODE, then it's pretty high up on the list, topics, and that sets the idea out. I tried to write those in a way, well, my dad helped me write those a lot, and he was a GP, but he was a coal miner, and then he became a GP. Back in the days where there was social mobility, but to read it so that they could understand it, and so that people could understand it, because obviously it's a complex idea. And I think I tried. Hopefully, they worked. I mean, I would read those two to start with, or my Twitter account.
Cool. Yeah, I'll
just not to put too much on there. I don't use it that often. I'm not a heavy user, so it's quite light.
Oh, great. I'll put some of those links in the description. So yeah, thank you,
the
Ben.
other thing is, sorry, one more thing on that. So a lot of these animations, because one of the big things I'm trying to describe this, and I'm doing this sort of useless hand actions. One of the things we realized was to actually visualize this, we should make animations of what's going on. So with Sam Barnett, who was who wrote the MeshCODE scale with me, we actually made a series of animations which are on my YouTube channel, which show what these memory molecules look like and what memory written in the shape of a molecule looks like. So that's quite good, because they're only like 15 or 20 seconds long each. So, but they just show a specific thing which we're interested in. So that's another good place to look.
Yeah, I can vouch for that because I watched them myself and they're very good. So I'll link that too. So yeah, thank you, Ben, for coming on. Yeah, it's been a really great conversation, and I'm super interested to see some of your publications when they come out, and learn more about this MeshCODE theory.
Brilliant. Thank you, Eleanor. That's been a really excellent chat. It's good.
So I hope you've learned something in this video. Thank you to my Patreon supporters, and thank you for listening.